Hello friends, and welcome in another video lecture of Easy Life Science. Here I am talking about some important questions, answers and explanations which are frequently asked for a SET Life Science exam part 4th. So please subscribe my channel Easy Life Science, like my video, share it to your friends, and don't forget to press the bell icon so you will get the notification of my newly uploaded video. So let's start. Here I am starting from a question number 126. Question number 126 is plasma represents dash dash percent of the total blood volume. The answer of it is option C, that is a 55. Plasma represents 55% of a total blood volume. The blood plasma is a light umber colored liquid component of a blood in which blood cells are absent, but it contains proteins and other components. It makes up about 55% of a body's total blood volume. Important elements in a plasma includes albumin, coagulation factors, fibrinolytic proteins, immunoglobulins, and other proteins. So option C is the correct option for question number 126. Then question 127, a serum differ from a plasma in that. Serum is a plasma minus fibrinogen and a clotting factor. Option A is the answer of question number 127. The serum is a blood plasma without clotting factor or a blood cells. The clotting factor like fibrinogen and other clotting factor are absent in a serum. Serum is a liquid that remains after the blood has clot. Plasma is the liquid that remains when a clotting is prevented with a addition of an anticoagulant. So this is a serum and this is a blood clot. In this serum, there is no any presence of a clotting factor or a cell. This is another image which shows a differences in a serum and a plasma. Then question number 128, hypoproteinemia produces edema by. Hyperproteinemia, it is a decreased serum protein condition. This condition reduces osmotic pressure of a blood, reducing a loss of fluid from intravascular compartments or the blood vessels to the interstitial tissues and resulting into a edema or a edema. Whereas the hyperproteinemia means the increased level of protein in your blood plasma. This level is higher than the normal level. Plasma is a liquid part of a blood. There are two main blood proteins, albumin and globulins. Hyperproteinemia may be seen in dehydration due to the inadequate water intake or to excessive water loss during vomiting and diarrhea, or a result of increased production of a protein. So here question is asked for a hypoproteinemia produces edema by. It is because of decreasing colloidal osmotic pressure across the capillary wall. Option C is the answer of this question number 128. Question 129, viscosity of blood increased with rise in albumin level. The blood viscosity is measurement of the thickness and stiffness of individual's blood. It is a direct measure of the ability of blood to flow through the blood vessels. Blood viscosity increased by a deformity of shapes of RBCs. So option A, that is albumin level, increases a blood viscosity. Question number 130, which of the following promotes Rolex formation? The fibrinogen promotes Rolex formation. The Rolex formation is lining of RBCs into chains resembling stacks of coins. Here is a Rolex formation of RBC. The RBC are in a chains resembling a stacks of coins. Some Rolex is normal in a dogs and more occur in a normal cat. Increased Rolex formation in a canine is associated with an increase in fibrinogen or acute phase proteins and usually seen in inflammatory diseases. So option C is the answer of question number 130, that is a fibrinogen. Then question number 131, hemoglobin has dash times affinity for carbon monoxide than oxygen. The hemoglobin has 250 times more affinity for carbon dioxide than oxygen. The carbon dioxide has a greater affinity for a hemoglobin than oxygen and a CO2. When carbon monoxide is inhaled, it binds to the hemoglobin to form carboxyhemoglobin, which is about 250 times more stable than that of the oxygen hemoglobin complex. Hemoglobin has less affinity for CO2 as compared to O2 because the CO2 is a larger molecule than oxygen. CO2 changes affinity of O2 to the hemoglobin. When a CO2 is in a blood, it reacts with water to form a bicarbonate and hydrogen ions. As the levels of CO2 in a blood increases, more H plus ions are produced and pH decreases. This increase in a CO2 and subsequent decrease in a pH reduce the affinity of hemoglobin for oxygen. So option C, that is 250, is the answer for question number 131. Then question number 132, iron content of hemoglobin is, the iron content of hemoglobin is 0.30%. Old RBCs are destroyed in lungs, liver and spleen. Option D is the answer of question number 133. RBCs are found in a blood. They are formed in a red bone marrow. The lifespan of RBC is 120 days, after which they are destroyed in a spleen. The spleen is called as a graveyard of RBC. Liver, lungs, kidney, these are the organs where the RBCs are destroyed through the process called as a hemolysis. Myeloid phagocytic cells in spleen, liver, lungs, kidney destroyed the old RBCs through a hemolysis. So option D is the answer of question number 133. Question number 134. Tissue microphage system includes alveolar microphage in a lungs, Kupffer cell of a liver, osteoclast in a bone. Now about the Kupffer cells of the liver. Kupffer cells are also known as a stellate sinusoidal macrophages, and these are the macrophages found in sinusoids of a liver. In fact, Kupffer cells make up 80 to 90% of all macrophages present in entire human body. Alveolar macrophages or AM, also known as dust cells. These are types of WBCs. The alveolar macrophages are the first line defense against invading respiratory pathogens, and osteoclast present in a bone are large multinucleated phagocytic cells derived from the macrophages, macrophage monocytic cell lineage. Osteoclast degrades bone to initiate a normal bone remodeling. So option D is answer of question number 134. All of them are tissue macrophages. Most common form of thalassemia is, the most common form of thalassemia is beta minor. Thalassemia is an inherited blood disorder characterized by a less oxygen carrying protein, that is hemoglobin, and fewer RBCs than a normal level. So option B is the correct option for question number 135. Then question number 136, thalassemia is characterized by deficiency production of alpha and beta chain. Question number 137, in sickle cell anemia, the defect lies in which polypeptide chain? The anemia is a result of a mutation in a single nucleotide. It is called as a point mutation in a gene, that is a HBB gene, a hemoglobin beta gene found in a chromosome number 11th, P15, that is a short arm of a chromosome number 11. It is autosomal recessive disease. So the answer of question number 37 is option B, that is a beta chain. Here hemoglobin is made up of 4 chains, 2 alpha and 2 beta. In a sickle cell anemia, there is a point mutation which causes replacement of a glutamic acid to a valine in a beta chains of a hemoglobin, and it results a abnormal hemoglobin. This is a abnormal hemoglobin. It is because of a point mutation where a glutamic acid in a beta chain is replaced with a valine. So it is a abnormal hemoglobin. This is a normal hemoglobin. The abnormal hemoglobin under low oxygen conditions in a RBC, the RBC got a sickle cell type of shapes, and these sickle celled RBC blocks small vessels producing a microvascular occlusions. Here we can see for a microvascular occlusions which stops blood flowing to the further points, and so that there is a necrosis of tubules and it causes a death of that tissue. This is about the sickle cell anemia. The sickle cell anemia, it is because of a mutation in a beta chain. So option B is the answer of this question, question number 137. The next question 138, average mean corpuscle diameter is, it is 7.3 micrometer. MCHC mean corpuscle question number 139, average volume of RBC is, the average volume of RBC is 86 cubic micrometer. Question number 140, average RBC count in adult male is 5.5 millions per millimeter cubic. Question number 141, MCHC is, it is a average amount of hemoglobin in a single RBC in a percentage. MCSC, mean corpuscular hemoglobin concentration, is a measure of average concentration of hemoglobin inside a single RBC. The range of MCSC is 32 to 36 grams per deciliter. So, option C is the answer of question number 141. Question number 142. Normal range of MCSC is option C, that is a 35 plus or minus 3 grams per deciliter. Then question number 143. What is the maximum concentration of a hemoglobin normally found in a RBC? The hemoglobin concentration normally found in RBC is 38%. Question number 144. Erythropoiesis is completed in dash dash day. It is completed in a 7-day. Erythropoiesis is the process which produces a erythrocyte or a RBC, which are developmented from erythropoietic stem cells to a mature RBC. It takes 7 days. Option B is the answer of question number 144. Question number 145. True about a bilirubin. Clinically, jaundice occurs if a level is more than 2 mg per 100 ml. Yes, it is correct. There is a jaundice when there is a high level of a bilirubin than 2 mg per 100 ml. Bilirubin is an orange yellow pigment that is produced by a breakdown of RBCs. Liver takes the bilirubin from a blood and changes its chemical makeup so that most of it is passed through a poop as a bile. If bilirubin levels are higher than normal, it's a sign that either RBCs are breaking down at unusual rate or liver is not cleaning a bilirubin as a waste properly. Normally, the bilirubin level is 0.1 to 0.2 mg per deciliter. The bilirubin produced by macrophages, it is a orange yellow pigment. After a breakdown of RBC, the liver takes it through a bile duct and cleans it as a waste. Then the next question, question number 146. Neonatal jaundice normally appears on a second and a third day of life. Option C is the answer of it. Question number 147. Clinically, jaundice appears in infants when a serum bilirubin rises beyond 5 mg%. Option D is the answer of it. Question number 148. Which is not a physiological cause of leukocytosis? Newborn, trace and exercise, these are the causes of leukocytosis. The leukocytosis is a increased level of WBC, whereas a leukopenia is a decreased level of WBC in a blood. Newborn, trace and exercise causes a leukocytosis. The pyogenic infection does not cause a leukocytosis. So option C is the answer of question number 148. Question number 149. All may be cause of a leukopenia except. Leukopenia means decreased level of WBC in a blood. Bone marrow depression, typhoid fever and starvation, these are reasons for a leukopenia, but early mornings hour are not a reason for a leukopenia. Then question number 150. Not true about a leukemia is. The leukemia is a cancerous condition of blood. Yes, it is associated with a immature WBC in a peripheral smear. It is correct. Associated with a tremendous proliferation of myeloid series of cells in a bone marrow. It is also correct, but it is not associated with a leukocytosis because a leukemia is associated with a leukopenia. So option B is the answer of question 150. Then question 151, thrombocytopenia is commonly occurs in. A thrombocytopenia is a condition where there is a low amount of platelets in a blood. The platelets or thrombocytes are a colorless blood cells that help for a blood clotting. It is a major symptom in a dengue fever. Thrombocytopenia is a major symptom in a dengue fever. A platelet count is less than 1 lakh per microliter is a severe level seen in a dengue fever. Normally, the platelet count is in a range of 1,50,000 to 2,50,000 per microliter of a blood. Then a next question in a purpura. In a purpura, 4 options are there. Platelet count may be low, capillary contractility is defective, CT is normal but BT increases, all of the above. The purpura, also called as a blood spot or a skin hemorrhage. Purpura are also called as a blood spot or a skin hemorrhage. Purpura occurs when a small blood vessel bursts, causing a blood to pool under the skin. Normally, the BT, that is a bleeding time, is 2 to 7 minutes, and CT, that is a clotting time, is 8 to 15 minutes. In a purpura, blood count may be low, CT is normal but the BT increases, and capillary contractility is defective. So all of above occurs in a purpura. So option D is the answer of this question. Then question number 153, carbaminohemoglobin is formed when a CO2 reacts with a hemoglobin. Carboxyhemoglobin is formed. Then question number 154, hemophilia is a X-linked recessive disease. It is a disorder in which blood cannot clot properly. Excessive bleeding occurs after any injury or a damage. It is a genetic disease. It is a X-linked recessive disease. Two main types of hemophilia are there: hemophilia A and hemophilia B. Low amount of clotting factor 8 in a hemophilia A, whereas low amount of clotting factor 9 in a hemophilia B. So option C is the answer of question number 154. Then question number 155, blood group antigens are found in saliva. Option D is the answer of it. Rh antigen is a D. Option D is the answer of it. Question number 157, true about a Rh factor. Against a Rh factor, there are no any naturally occurring antibody found in a rhesus monkey. So name is given as a Rh factor. Option A is the answer of it. Then question number 158, ESR is a erythrocyte sedimentation rate. Option D is the answer of it. ESR is determined two methods. One is a Wintrob's method and Westergreen's method. So answer of question number 159 is option C, both A and B. Then moving to the next question, question number 160, transitional epithelium is found in. The transitional epithelium are found in a urinary bladder, urethra, or a ureters. Transitional epithelium, also known as a uretholeum, they are present in lines of urethra, ureters, and urinary bladder. So option D is the answer of it. In question number 161, histamine secreting cells are found in a connective tissue. Option A is the answer of it. Question number 162, characteristic of a simple epithelium is that they make a definite layer. Option C is the answer of it. Question number 136, areolar connective tissue joins a histamine. Ignore this question Then question number 164. Hallucination cannot occur in a large bones. The hallucination system or a canal are a longitudinal canals that run in the walls of a long bones like femur and humerus. They contain narrow fibers and one or two capillaries. The long bones like humerus and femur shows a Haversian canals. So option A is the answer of question number 164. Then question number 165. The surface of simple squamous epithelium is. The surface of simple squamous epithelium is silitably permeable. So option B is the answer of it. Thank you for watching my video. Please subscribe my channel Easy Life Science, like my video, share it to your friends and press the bell icon so you will get the notification of newly uploaded video. So thank you once again.